We spent a good amount of time understanding prokaryotes and eukaryotes, and one of the organelles we talked briefly at that time was mitochondria, and I said it was the powerhouse of the cell. In this particular chapter, we will discuss many activities that takes place within this organelle, and in the absence of this organelles, how do microbes live? The major events that take place for a living cell includes many complex biochemical reactions. These reactions are much more intense and more complex in a eukaryotes than in a prokaryotes. However, the microbes do need to have energy provided to it or a continuous source of energy provided for all these reactions, and these reactions are hence called as metabolic reactions. So what is metabolism? Metabolism is a term that refers to the sum of all chemical reactions and activities that take place within a living organism. These reactions can be categorized in two categories. One is catabolism and the other is anabolism. Catabolism and anabolism, you need to know the definition of each. Catabolism involves breakdown of complex organic compounds into simple ones. So in catabolic reactions, larger molecules will be broken down through reactions that are called as hydrolytic reactions. Hydrolytic reactions are those, or hydrolysis are reactions in which water molecule is used to break the chemical bonds. These reactions are also exergonic. Exergonic means that they produce more energy than they consume. Hence, you see in this reaction over here that ATP plus phosphate is required to break these molecules down, and they will produce more energy than they consume. So that's why you see the production of ATP up here. An example of catabolic reactions, for example, would be the breakdown of sugar into carbon dioxide and water. Some of you have done these reactions in other biology classes, so we're pretty much reviewing the same kind of reaction. The other category of reactions that are anabolic reactions. Anabolic reactions are also called as dehydration reactions. These are reactions in which the complex organic molecules are synthesized from simple molecules. So anabolic reactions, they will transfer energy from ATP to the complex molecules. These reactions are called as endergonic reactions. So I'm going to go ahead and write down in the text over here, these are exergonic as they release energy, and these reactions, which are the anabolic reactions, are endergonic as they consume energy. So once you can differentiate the two kinds of reactions, it will be very difficult to look at, and very easy to look at a reaction and determine whether it's exergonic and endergonic. One of the easiest thing I tell students to look at is notice the way the molecule of phosphate is written over here. If actually I was trying to, that didn't work, I was trying to highlight it. So energy, if it's written to you on this side of the equation, that is on the left side, that this means that this reaction is going to use energy. And then if you see energy on the other hand, that is shown on the right hand side, it means that this reaction is going to release energy. Okay. Both of these reactions are categorized by or controlled by enzymes. and these enzymes are very unique, that each set of reaction has many steps, and each step is controlled by one particular unique enzyme. Here is a complex picture that's shown to you on the slide, and it's actually it's a summary of all the reactions. Notice the complex molecules such as starch, proteins, and lipids are shown to you on the bottom. Catabolic reaction will transfer the energy from these reactions to, that means it will break down the energy from the larger molecules to smaller molecules. So you have the smaller molecules that are glucose, amino acids, glycerol, and fatty acid. On the other hand, there are reactions that will take these smaller molecules and transfer the energy back into the larger molecules, and these release energy. So both of these reactions are complementing each other. Both of these reactions are going to be what is called as coupled reaction, because by coupling we mean that as ATP is break down in anabolic reactions, and catabolic reactions are coupled to synthesize that ATP. Now let's look at in more depth as to the difference of the different reactions that take place within each category. Hydrolysis was a reaction that is a kind of reaction that undergoes in catabolic reactions. Hydrolysis comes from the word water, and lysis is breakdown. So literally in this word, what is happening is water molecule is being used, as you can see up here, a triangle showing water molecule is shown up here. Water is being used to break the linkage between these two molecules. So here is an amide linkage, a peptide bond, and these are the two AA1 and AA2, or the two amino acids. Water molecule is used to split the bond, so it breaks a bond. Notice the OH is attached to the carbon on this end, and hydrogen is attached to the NH, and it splits the two amino acids separately by the addition of hydrogen and OH to each individual or adjacent amino acids. So hydrolysis is going to be a reaction that is going to take place within catabolic reactions. Condensation or dehydration synthesis is just the opposite. Condensation, which refers to dehydration, that is removal of water molecules. Notice up here we have two glucose molecules that are adjacent. One OH is taken from one molecule, another hydrogen is taken from the other molecule, and water is formed and removed from this environment, and this oxygen is now linking the two glucose molecule. So this is a condensation or dehydration synthesis. I know that when I taught the previous biology classes, I have really emphasized my students with the difference between the two. So it's more important that you understand the difference between the two. Now, looking at exergonic and endergonic, can you reflect as to which one is which? Now remember, condensation is taking place in anabolic reactions. So catabolism process always involve endergonic reaction. That is, it will use energy to form. And hydrolysis reaction always takes place in catabolic reaction, so that's when the energy is being released, or exergonic. Now, we talked a little bit about enzymes. So what are enzymes? Because without enzymes that are needed for each of these reactions or each step, none of this metabolic process can go on. Enzymes are mainly, firstly, enzymes are all protein in nature, or majority of the enzymes are usually protein in nature. Not all proteins are enzymes, but remember, all enzymes are usually protein in nature. Now, the enzyme theory is based on the collision concept. Collision talks about two molecules that, when they react, they need to collide with each other, which collision literally means that they need to have energy that the particles of one will collide with the other. And when this collision takes place, energy is required. So that's the nature of a start of any kind of reaction. That energy that is required to collide is called as the activation energy. So there should be enough activation molecules for any reaction to take place. For example, if we have a cup of sugar that's lying on the table, there is enough potential energy within the sugar molecules, but nothing will take place unless the molecule are being activated. So when we say activation, what do we mean? Activation is a state when the collision energy is required. It is the amount of energy that will be required to disrupt the stable configuration. So for example, if I take a cup of sugar and put it in a beaker or heat it, then I'm applying or supplying energy to these molecules, which will rearrange the electrons. That is an activation energy. So number of molecules above this activation energy will require equals to reaction rate. So if more activation energy is there, then the reaction will take place. Enzymes are molecules that lower this activation energy. For example, in this diagram over here, if you notice in this diagram, the reaction without the enzyme is shown to you in this black region that I'm pointing my mouse to. Which would mean that these two molecules should have an energy up that is up to this level. The blue mark over here, if you notice, the arrow activity shows the amount of energy that's required for this reaction to start. With the absence of an enzyme, you will have to provide this much energy. However, if enzymes are present, then that lowers the activation energy and the reaction can go on with a lower activation energy. Thus, enzymes are always needed in a very small amount. They're catalyst. They do not get used up in a reaction. They only speed up a chemical reactions, and biological catalysts are called as enzymes. Each enzyme also needs a specific substance to work on, and that is called as the substrate. So for example, an enzyme which would only work on, for example, lipids would be lipase. An enzyme that will only work for a particular kind of lipid would be named after that substrate. So enzymes have many names, and usually the names of enzymes ends in ASE, and the names are based on the substrate that they're working on. So they change the name of, for example, sucrose to sucrase. Protein would be called as proteases. So usually you will find the names of the enzyme is ending in ASE. What does the enzyme work on? The enzymes will work on a substrate. Here in this model, we can see that there is an enzyme that's shown to you in blue. And notice there's a unique shape over here. The purpose of shapes is to represent the way the chemical bonds are present in that molecule, which gives it a unique arrangement. Now, if various substrates are present, then the only substrate that can fit into this region of the enzyme will be able to work with this enzyme. This unique region where the substrate will work is called as the active site. Active site is a very important region. It's just the region where the enzyme will form a complex with the substrate. Now, just like you have, we all have keys, and the right key will fit in the car or any lock that we own. So that is the concept of a lock and key model. So in other words, we're talking about the lock being the enzyme and the key is the substrate. So when you have the right substrate, then it will open up that lock. So that is the lock and key model. The enzymes are highly specific. Each unique shape will be fitted by a unique enzyme. That is, the active site on the enzyme is unique to a substrate. Lots of times you have generic enzymes, for example, dehydrogenases or ligases or hydrogenases or transferases, and these are groups of enzymes that catalyze a kind of reactions, but they are unique with the substrate that they work on. Now, this little diagram that is shown to you over here, or the model, I will ask you in a test to draw it out. So make sure you know how to draw this model out. As I said earlier, the names of the enzymes are going to end in ASE, as we can see over here. The turnover number is the number of molecules that the enzyme will convert per second. So, for example, DNA polymerase the number is 15, and lactase dehydrogenase is 1000, which means that it doesn't get used up. It works on a certain molecules, and then it can be reused. So the higher the turnover number, that means it can work on a lot of different molecules of the same kind of reaction. Okay, let's look at the parts of an enzyme. Mostly the main part of the enzyme is a polypeptide chain, which I said to you over here is a protein. Let's write down over here. This polypeptide chain is called as the protein chain. Besides the protein, there are other parts of an enzyme. The apoenzyme is the polypeptide chain of the enzyme, and then it will have two parts beside that, and one is called as a cofactor or a coenzyme. Now, cofactor refers to inorganic molecule, and coenzyme is an organic molecule. So, for example, when we talk about vitamins, vitamins are coenzymes. They're organic molecules. Cofactors would be, for example, calcium ions, zinc ions, manganese ions. Those are cofactors. So here is trying to get this. This should be, need to move this so you can see better. Okay, nicotine adenine dinucleotide. This is the name of an enzyme which is usually present in electron transport chain. Nicotine adenine dinucleotide phosphate is another enzyme, and they're all present in these metabolic reactions. NAD is used up in catabolic reactions, and NADP would be in anabolic process. Another enzyme is coenzyme A, which is a derivative of vitamin B, and in particular it's called a pantothenic derivative, and that's obtained from niacin. Metals such as copper, magnesium, manganese, zinc, calcium, and cobalt are all going to be cofactors. So together, the apoenzyme and the cofactor or the coenzyme make up which is called as a holoenzyme. So here is a holoenzyme when you have the enzyme, the cofactor, and the substrate together. So all of this together will make up the holoenzyme or the holoenzyme. Now, enzymes react the mechanism by which the enzymes will work is based on two processes. One is called as competitive inhibition, and one is called as non-competitive inhibition. Now, inhibitors are pretty much molecules that will affect the growth of an enzyme. It will really inhibit means to slow down or to totally stopped. So they have competitive inhibition, which means that these inhibitors fill the active site of an enzyme and compete with the normal substrate for the same active site. Non-competitive inhibition is that they do not compete with the substrate for the enzyme active site, and instead they will interact with another part of the enzyme, and or they will actually that's actually an allosteric site that they will block another site on the enzyme, which is called as the allosteric inhibitor. In the non-competitive inhibitor, sometimes I usually give best examples to remember these. For example, poison would be a non-competitive inhibitor, because in poison, for example, cyanide, or they can bind with the iron-containing enzymes, and fluoride can bind with the calcium or magnesium, and then they actually block an allosteric site, another site that is away from the active site, and they distort the shape of the enzyme, and the substrate cannot fit in. Notice that the shape over here is distorted. In competitive inhibition, for example, various antibiotics, that's how they work. Certain drugs, sulfur drugs, and para-aminobenzoic acid, which is PABA, these molecules, they may resemble the true substrate, but not exactly is the true substrate, and they can bind to the active site on the enzyme and thus block that enzyme's growth. So, for example, when we take an antibiotic, the purpose of the drug is to fool the enzyme. So when you take that for a 10-week duration and you take a certain amount, then you are actually destroying the enzyme activity of that microbe. And eventually, when there is no enzyme activity, the metabolic process will decline and the growth will stop. There are certain factors that will affect enzyme activity, and enzyme activity will actually, these factors, they destroy the enzyme's activity, and they destroy that because they denature the enzyme. Denaturing an enzyme means breaking down its three-dimensional structure. So, for example, up here we have this enzyme that is more complex and lots of bonds and spiral arrangements. Temperature or heat are two factors or pH will affect the three-dimensional structure of this enzyme, and it will denature the protein. Here is a graph that represents the factors influencing enzyme activity. For example, let's look at temperature. Notice that the range of temperature is between 35 to 42 or 41. You have the maximum activity. If you increase the temperature or you decrease temperature, you're slowing down enzyme activity. pH, again, we have a bell curve. Nice bell curve shows between 4 to 6 is your maximum pH, optimum pH for enzyme activity. Any decrease or increase will lower the enzyme activity. The substrate concentration is shown over here. Notice that even if you increase the number of enzyme, it's not going to do much to the speed of the reaction, because if there are only 10 molecules of a substrate and you have 3 enzymes, and that's fine. They can work slowly and then they always get reused. However, if you increase from 3 to 10, you may actually be wasting the enzyme molecules, because the substrate, the enzyme, all the substrates are caught up and you don't really need more enzymes. So it will be a plateau after that. I think we already did this. Let me see. Yeah, I should have this over here. It's the same thing. This is in competitive inhibition. No, actually, this is a little different. This is competitive inhibition. The image looks very similar to that one. I should have moved this slide earlier. In competitive inhibition, notice that the normal binding of substrate versus the enzyme inhibitor. Here is the inhibitor that is blocking the activity, and the true substrate cannot compete with the active site. So this is competitive inhibition. The example, for example, I gave you was the antibiotics, where it is blocking the active site. And lastly, we want to look at a feedback inhibition. All allosteric inhibitors play a role in a kind of these reactions, which are called as feedback or end product inhibition. In this mechanism, the process is stopped when making of a substrate based on the end product. For example, if you look at this substrate over here, we have the substrate, and the pathway operates at various different steps. In each step, you have one enzyme. Now, if you block the allosteric site, for example, on this enzyme, then the pathway will shunt down because it will not give signals. Now, if the pathway was to correct, for example, it goes all the way to the end product, the release of this end product, this small molecule that is shown over here in orange, notice it goes back and it blocks this enzyme, and it will block the substrate. It will block the enzyme. It will pretty much shut down the substrate. This kind of inhibition it requires several steps, and the synthesis of a particular chemical compound, which is the end product at the end, is like an assembly line. For example, each enzyme or each step is catalyzed by a separate enzyme. Enzyme 1 is catalyzing intermediate A, enzyme 2 is intermediate B, enzyme 3 is the last process. In this process, the final product can inhibit allosterically the activity of one of the enzymes earlier in this pathway, and this is called as feedback inhibition. Feedback inhibition generally acts on the first enzyme in a metabolic pathway, because once this first enzyme is inhibited, the product of the enzyme's reaction is not synthesized, and the unsynthesized product would normally be the substrate for the second enzyme in the pathway, and the second enzyme reaction will stop immediately. Bacterium E. coli uses this feedback inhibition in the synthesis of the amino acid isoleucine, which is required for the cell's growth. For example, if isoleucine is added to the medium for E. coli, it inhibits the first enzyme in the pathway, and the bacteria will stop synthesizing the enzyme. And this condition will be maintained until the supply of isoleucine is depleted. And this regulates the production of other amino acids such as vitamins and purines and pyrimidines. It's a natural process for the metabolic reactions not to get overused. So this concludes our first section on metabolism, and we will continue to the next section.